Good morning, everyone, and welcome to this analytical chemistry class, where we'll be carrying on our discovery of instrumental analysis by having a look at some of the components inside of optical instruments. So last time we talked about things like waves and electromagnetic radiation, how they interacted with matter, and some things like that. So now we're going to have a look at some of the components that go into the design of these instruments and in some way tailor their response so that we can actually get meaningful results out of it. So like which kind of light source or radiation source do we use for which kind of spectroscopy, or which kind of transducer do we use, which kind of optical components do we make use of. So usually whenever we're doing these kind of optical measurements, there are 6 major forms of phenomena that we make use of. So they are absorption, fluorescence, phosphorescence, scattering, emission, and chemiluminescence. So absorption is the one we'll focus on the most. That's the idea that our molecule or our compound will absorb a certain wavelength of light. Usually what's going on during this process is some form of excitation from a ground state to a higher excited state, whatever that excited state may be, whether it be electronic, vibrational, or rotational. We'll mostly be considering electronic excited states, but others exist like vibrational and rotational. Fluorescence is whenever we have emission of light from a compound after it has been excited to that excited state. So that's where it's different from emission. Emission is kind of spontaneous, whereas fluorescence is after some sort of absorption event has occurred. And then phosphorescence is similar to fluorescence, just on a longer time scale. Scattering, we've already talked about in the previous sessions, where light is scattered by some particles or something. Emission is generally spontaneous, and chemiluminescence is due to some sort of chemical interaction or reaction. So in all cases, response is proportional to concentration of the analyte. So the general design of these optical instruments that we'll be talking about. So many optical instruments share similar designs. So they have a stable radiation source. So like the light that comes from the we need some sort of radiation, basically. Whether that just be probing radiation or whether that radiation be used for excitation in the case of absorption measurements or in fluorescence measurements, it needs to be stable. What does that mean? It means that like it produces a known range of wavelengths. So whether that be from, I don't know, 200 to 1,000 nanometers, basically it's consistent. And the amount of intensity of that light doesn't fluctuate too much, because that can be problematic in maintaining how many molecules absorb light. So for example, if you increase the intensity, you have more molecules absorbing the light. If you decrease the intensity, you have fewer. And so that changes the output of your instrument. Like the readings you get could change. And so that would be problematic on like measuring a large number of samples. Transparent sample holder. So this transparent doesn't necessarily mean that it's transparent to our eyes. It just means it's transparent to the wavelength range that we're measuring. So if we're measuring in the UV or in the visible or something like that, usually it means it's transparent also to our eyes. But the most important thing is that it doesn't have an absorption or an emission in the wavelength range that we're interested in. Then we have some sort of wavelength selector. This is important to kind of like make sure that we're not measuring the light intensity from wavelengths which are not meaningful to our measurement. We have some sort of radiation detector. So this can be like a transducer type thing. Usually it's like a photodetector of some sort. And then we have a signal processor and readout. So we'll be focusing on the first 4. The last one is kind of just a computer effectively, and we won't be considering that, but be aware that there are multiple components. And this, we've seen this before. We saw this last time, this typical absorption setup where we have our light source here at the start. We have a parallelizing lens, which takes this sort of diffuse incoherent light source and turns it into a more coherent form. And then we have a filter here of a shutter, which can turn off and on the light without turning the lamp on and off, because these lamps are typically quite fragile. What that means is like they don't respond well to changes in temperature. Like, for example, if you turn it off and on, you're liable to destroy it. Then the diaphragm is sometimes there, sometimes not. Then we've got the sample cells, which we'll talk about today, photoelectric devices, and then the digital meter. So today we'll talk about the tungsten lamp, we'll talk about the filter, we'll talk about the sample cell, and we'll talk about the photoelectric device, but we won't talk about the readout. So as well, recall that a photoelectric device is a transducer that converts non-electrical signal to electrical signal. So that's important thing that we talked about before in the previous sessions, was that this idea of us actually recording or measuring the changes in electrical current or potential that flows through a circuit, as opposed to actually directly measuring, like, for example, the light absorbed. It's all by kind of inference and indirect measurement, because it's much easier for us to measure electricity flowing through a circuit than it is the actual amount of light, which is absorbed by some sort of photoelectric device. So we design it in such a way that it's easier for us to manipulate. So first, we'll start at the beginning of our absorption instrument, and we'll talk about the light sources. Now we'll talk about tungsten lamps, but there are also others available. So here are a bunch of different radiation sources and their spectral regions. So we learned about spectral regions last time, but we have the vacuum UV, we have UV, we have visible, we have near IR, we have IR, we have far IR. So these are the major regions that mostly we're interested in as analytical and instrumental chemists. So typically UV vis is very, very useful in quantifying the amount of something that is present. And so that's something that we'll probably come across a lot if we work in or around a company which is either making a product or is selling a product which has some sort of chemical component in it. So UV range and visible range, whilst they're beside each other, it's not necessarily true that the light sources are the same. So for example, in the case of UV vis, usually we need what's called a continuum light source. So this means it produces a broad spectrum of wavelengths. So it produces a lot of wavelengths over the entire range, which is different from line type radiation sources, but I'll talk about that in a moment. So for example, usually in UV vis, you might want to use a xenon lamp. The problem with the xenon lamp is it's very expensive. So usually most modern UV vis spectrometers actually just make use of two lamps, one which is a hydrogen or deuterium lamp, usually deuterium because it's more stable, and a tungsten lamp. These two combined together give us the full range of the UV vis, and they don't break the bank. Then LEDs can also be used, but you can see that LEDs struggle to get down into the full range of the UV, which is problematic. I mean, what if you want to measure down in the lower range of, like, for example, it only goes from like about 300 all the way up through the visible into the near IR, but we're talking about UV vis experiments which combine both of these regions, but it doesn't cover like 200 to 300. So that's a problem. Same with the tungsten lamp. That's why we combine it with this H2 or deuterium lamp. So it's possible to combine this with LEDs or tungsten lamps. The rest are kind of specialist or a little bit older in use. They typically don't see a great deal of use. Now, nichrome wire sometimes is seen, but that's not for UV vis measurements. So UV vis measurements are typically these four here. Then the line sources, so what they do is they produce a very narrow, so Continuum produce a lot of different wavelengths all at the same time. Line produces very few wavelengths. So they're very specific to a certain spectral region. So whilst it says here, for example, that lasers cover this entire range, that's because you can tune a laser to produce wavelengths over those different ranges. It doesn't mean that the laser produces a wavelength over the entirety of this spectral range. It really just produces like little lines the whole way along, and you can tune it to be at different wavelengths. The most common, usually around 800 nanometers. Hollow cathode lamps, then, they also cover UV vis. These are line sources. We'll talk about those shortly. But they are used in one of the kinds of spectroscopy that we'll learn about next time, which is atomic spectroscopy. And that's because they produce wavelengths of light specific to specific elements. So for example, you have a particular lamp for each element that you want to measure. So you have an iron lamp for iron, you have a copper lamp for copper, you have a tungsten lamp for tungsten, that kind of thing. So detectors then, we'll talk about those later on. The most common ones that we'll be using are photon detectors. So like photomultiplier tubes, phototubes, photocells, charge transfer detectors, photoconductors, things like that. But we'll talk about those later, silicon diodes, what have you. We'll talk in detail about some of these, the most common basically. These thermal detectors are mostly for infrared measurements, which we won't talk about in this course. Light sources then, okay, the continuum light sources. So as I said, continuum sources produce a broad range of wavelengths, and sometimes they're black body radiators. UV lamps have a broad continuum of wavelengths with consistent and stable radiant power across the full range. So it doesn't mean that the power is the same over the entire range. It just means that they produce enough power over the entire range for it to be useful. Then, so for example, as I said, deuterium hydrogen lamps produce continuum UV light, and so they're typically made use of, but D2 lamps are preferred because the output is larger and brighter than the H2 lamps, the hydrogen lamps. So this is what one of those light bulbs looks like. You don't really need to remember what it looks like, but it's just what it looks like come out of the textbook. And then this is the spectrum for those lamps, those H2D2 lamps. And you can see here that they produce light over a broad spectrum. So you can see it goes from around about just over 200, about 210, all the way through to about 400 nanometers. But you can see the intensity here is not consistent. It's not the same. So the intensity is higher at certain points and lower at other points. But the important thing is that it does produce light of sufficient intensity over that entire wavelength. And so then we have the ability, I mean, it's consistent. It does that consistently. And so that consistency is useful because what it allows us to do is to make sure that our instrument knows that and so can respond to that so that it can kind of like correct for that lamp-induced error. So it can kind of try and create a consistent spectrum. So instead of like, so it might amplify certain parts of the spectrum so that it's not being manipulated by the presence of these changes in intensity. And sometimes it's not necessarily necessary, but it depends on the system. But your instrument is usually capable of doing that, especially modern instruments. Components, sources for visible light now, we have tungsten lamps. These have a range between 350 and 250 nanometers. They really are just resistance lamps. So you pass an electrical current through them, it heats up, produces light. A bit like the old style light bulbs, not so much now, but before, maybe like 10 or so years ago. Tungsten halogen lamps, these are a development of tungsten lamps. So they're pretty much the same. They produce the same sort of range, but they have a presence of a halogen inside the lamps, usually iodine. And this helps to increase the lifespan. Basically what happens here is that in the tungsten lamps, over time, the tungsten heats up and actually evaporates. But in the tungsten halogen lamps, because of the presence of the iodine, what happens is the tungsten heats up, evaporates, then reacts with the iodine, and then will deposit again onto the filament, which is what produces the light. So it somehow increases the lifespan. So the operating temperature then is increased to 3,500 Kelvin. So these things are ridiculously hot. And then this is sort of like one of the wavelength intensities which can be obtained from these sorts of lamps. And so less common sources are LEDs, semi-monochromatic, and more environmentally friendly, and xenon arc lamps, which have a range of 200 to 1000 nanometers. So LEDs, not that commonly, it's really mostly actually just H2D2 lamps and tungsten filament lamps, even until now. It's worked very well, and people are reluctant to change, basically. But there are some which make use of LEDs, and there are some which make use of xenon arc lamps. The problem with xenon arc lamps is that they're quite fragile and expensive. So they don't last as long, and then when they break, it's hard to replace them, or well, expensive to replace them. Line sources then. So line sources produce a few discrete wavelengths. So instead of producing the broad spectrum, like the 250 to almost 2000 nanometers produced by the tungsten lamp, here we have maybe a range of about 5 nanometers produced in some instances. Like it's a very, very narrow range of wavelengths that are produced. In the case of hollow cathode lamps, it's actually so specific, it's basically just one wavelength. So hollow cathode lamps are made of the analyte element. So inside of the lamp is some, like say this is for the detection of iron, iron as an element. Inside this lamp, there is some iron. And so what happens is the iron gets excited and it releases light of a specific wavelength, which only will be absorbed by iron. So that means that even if your sample contains other things like carbon or copper or I don't know, whatever, the only thing which will absorb will be the iron. And so the only thing you will be measuring is the iron. And so it's very powerful in that sense. It does, however, mean that the only thing you can measure is iron. So it's a two-edged sword. Like on one hand, you're very specific to one type of element, but on the other hand, you can't measure anything but that element. But sometimes that's all you want to measure. And so that's okay. Like this is very useful for environmental analysis. So if you're involved with like, I don't know, like checking pollution or making sure that businesses are compliant with the local laws and not discharging hazardous waste into the water table or whatever. You can collect samples and you can measure them using these sorts of hollow cathode lamps. For the particular things that you're interested in. Maybe you think they're dumping too much copper into the local river. You can take river water samples nearby the factory and find out. So tungsten anode, then there's the this hollow cylindrical cathode, which is made of the analyte element. Then there's a glass tube filled with neon or argon, as it says here. And then we have a specific lamp for specific elements. We'll learn a lot more about like the use of these in the next session when we tackle absorption spectroscopy. Then the cathode shape here is cylindrical and it focuses the beam in one direction. So it becomes basically an emission beam, which can be targeted towards whatever it is that we want to measure. And each atom absorbs at different wavelengths. So to analyze that element, light from that element is needed. This is because if we recall back to our previous session where we talked about the idea of there being two states. There's the ground state and then there's the excited state, and we can have an electron down here and it can be excited up to here, but this has an energy difference between it, like between here and here. So between the ground state and the excited state, there's an energy difference E. But that can correspond to a specific wavelength due to Planck's relationship. So that corresponds to a specific wavelength of light. And so if we have a hollow cathode lamp, this produces light of a specific wavelength to that element. And so we can measure only that, or like we can measure specifically that element. So produce, say, I don't know, let's just ballpark figure like 400 nanometer light for an iron atom. Then like anything else, like copper maybe absorbs at 450 nanometers. It won't absorb the 400 nanometer light. Same for something maybe like sodium, which maybe absorbs at 300 nanometers. It won't absorb the light either, because even it needs to be specific, like it needs to be actually like exactly the same, or else it won't absorb. And we won't get the absorption effect. Lasers we talked about, so we'll talk about very briefly here. We won't go into a huge amount of detail with these. They are reasonably complicated, but they produce very narrow wavelengths of light, and they're very, very reliable in terms of their energy output, consistent for hours and hours on end. If you've set it up correctly, like you'll get the same energy output for basically forever, so long as you have enough power and you have enough cooling and things like that. So laser stands for light amplification by stimulated emission of radiation. You don't need to remember that. You just need to remember that they're called lasers, but it does actually stand for something. So lasers produce light through two processes. One is what's called the pumping of an excited state. Basically, that just means that we're exciting a lot of electrons from the ground state to the excited state. So you remember this. So a lot of electrons go from the ground state to the excited state. Pumping just basically means that we're filling it up. We're increasing the population of this excited state. It's just a fancy way of saying it. And then the other one is what's called stimulated emission. So that's the SE part of lasers, stimulated emission. Basically, this is the amplification of the light intensity through constructive interference. So last time we talked about waves, and we talked about how they could interact with each other. And if they had the same sort of period of their wave, like these two, then they could interact constructively, and that would amplify the intensity. And so that would result in more intense light. And so that also happens in lasers. So they make use of two things. One is an increase in the excited state population. So there are more, so it has a process of trying to increase the amount of electrons in the excited state. And then it combines all of the emissions of the electrons to amplify the light intensity through constructive interference. Effectively, what the two processes are. And then this sort of like high intensity light of a specific wavelength is due to the presence of the two energy states within the lasing medium, whatever the material is that we're using. So it's not that dissimilar in a sense from the light that's produced from a hollow cathode ray tube, but the difference is that this one produces much more intense light, and it's not necessarily like one element for one species. Like, no, you don't just need, like, you can use the same laser for multiple different elements or multiple different molecules. So it's another form of way of generating light. Not really used that much in absorption measurements, because absorption measurements typically make use of continuum sources, but it is a form of light generation, which is important in things like infrared spectroscopy. So some advantages of lasers include the idea that it is intense. It has a large intensity. That's because of that stimulated emission, so that constructive interference. It's monochromatic. That means it only produces really one wavelength of high intensity light. So it's a line source. It's pulsed. It can be pulsed or continuous wave. So you can create packets of light, much like photons, or you can have it as a continuous wave where it's continuous. It's coherent, which means that you can transport it over a large distance and it won't diverge, which is related to the last point, which is small beam divergence. So this is important for the use of a laser. Like, I mean, usually in optical components or optical instruments, the light has to travel over quite a large distance. So if it starts to diverge or starts to spread out and become more diffuse, then that's not very good. You need a tight focused beam for as long as possible. So the next thing we'll go on to talk about, that was light sources. The next thing that we'll talk about then is this, the components inside, like the various materials used for the cells that hold our sample that we're interested in measuring. and things like that. So some of them include things like for our UV vis region, which is here, which is mostly what we're interested in. Things like fused silica or quartz are kind of like the top of the line. The reason for that is that they're very transparent over that range. Like they can hold a large number of different materials. Like they don't react very much with like any kind of organic solvent or anything like that. So they really are the top of the line really for UV vis measurements. They are, however, very expensive. You're talking like easily north of $200 for one regular sample holder. So it's a little bit prohibitively expensive. You can use glass, but glass has a problem where it starts to absorb UV light below 300 nanometers. So you can only really measure from about 350 through the visible range. It is good for the visible range though, that's fine. Silicate glass can also be used. So these are two forms of glass. They're also quite common. Plastic cuvettes are not very good for anything except for maybe aqueous solutions. Like polystyrene, what do you call it? Cuvettes, plastic cuvettes, not very good. Sodium chloride, potassium bromide, these are also very nice. Lithium fluoride even. These are very nice materials also. They're very transparent. They have a wide range of transparency. The issue is though, of course, like they dissolve in water. So, you know, sodium chloride dissolves in water if you've ever made a salt solution. So the issue there is that it cannot be used for measurements of anything which is aqueous. So a lot of things are aqueous solutions. So we can't use these for it, well, but we can use these for things like organic solvents or other things like that. We also can't use them in highly humid conditions because they would partially dissolve in the highly humid conditions. And these are like single crystals of sodium chloride. So they are kind of transparent even to our eyes. But if they start to frost up because of maybe some of the moisture dissolving some of the surface sodium chloride, then they're not going to be useful anymore. But they're not as expensive as something like fused silica or quartz, but they have a limited range of uses. Wavelength selectors. So these are ways of, so we saw earlier the continuum sources, such as like the tungsten lamp and the deuterium lamp. So sometimes we don't want to excite our entire sample with one particular type of, or like with all the wavelengths at once. Maybe we just want to measure at one particular wavelength at a time. Maybe that's because of the setup of our instrument. Maybe our photo detector or our transducer isn't capable of measuring everything at the same time. And that's quite common. So what we need to do is we need to select the wavelengths. So we need to select a particular wavelength to measure at a time. So we have, like, for example, two major types. We have continuum and we have discontinuous. Continuum is by far the, well, actually, no, both of them are pretty. Continuum basically is like a prism. I don't know if you've ever used like a glass prism where like it splits the light into like a rainbow effect. So like you put it in like the sunlight and then it like produces like a diffuse dispersed ray of multicolored light from white light. So that's a glass prism. So you can actually use that inside of an instrument to separate the different wavelengths of light. A more common one now is like what's called a grating, which is basically just a finely divided set of a piece of sheet of metal with lots and lots of very finely divided lines. You know, you're talking about from 3,000 lines per millimeter up to 50 lines per millimeter. Hopefully, you know how big a millimeter is. It's not very big, but imagine drawing 50 lines inside there, or even 3,000 lines. It is possible. These can be used to disperse the light in a way, and what happens is the light kind of like interacts with the line and then gets, because the line ends up being of a similar size to the light's wavelength. And that kind of like disperses it, or like causes different wavelengths to be reflected at different rates. Then the ones that we'll be talking about down here, discontinuous, like for example, glass filters or interference filters. We'll mostly be talking about glass filters. These are very useful, actually. They cut out broad ranges of light. The reason why they're called discontinuous is because they don't block all wavelengths of light. They only block them over a certain range, but they're pretty good at doing that. So this is kind of what we're looking for in terms of an ideal output from a laser or radiation source. What we want to be measuring is a particular wavelength, which has quite a narrow effective bandwidth. So we have here the nominal wavelength that we actually measure. So even though, like, for example, there's this like peak shape, what we measure is the position at the maximum. So whatever this maximum value is going to be our nominal wavelength, or the wavelength that we measure. So say this is 400 nanometers, we would say that we are measuring light at 400 nanometers. Then the effective bandwidth is going to be the width of the peak at half of its maximum. So say this is 80% transmittance, we would measure it at 40, because half of 80 is 40. So half the peak height is, the width at half the peak height is the effective bandwidth. So the peak width at half maximum. So, okay, so we've gone and talked about our light sources. Now it's time to talk about the next thing, which are our filters. So let's talk about the filters. So, which are our wavelength selectors. So wavelength selectors, these are used to select for the narrowest range of wavelengths possible. So we have two types. We have filters and we have monochromators. So these are the two most common types, the two that we will talk about the most. This is obviously the light that we just talked about. We'll focus mostly on filters, and we'll talk a little bit about monochromators. So filters, we have two types of absorption filters. So this can even just be a piece of colored glass. If you've ever been in these are quite common to see in old buildings or even in churches, where you've seen like the stained glass windows or something like that. That's an absorption filter, actually. We can also actually get a colored dye and place it between two glass plates. That can work as an absorption filter. Then interference filters are also a type that is present. So what they do is they use the idea of destructive interference to try and reduce the lights at certain wavelengths. Then we have monochromators, we have echelette and prism type. So the prism type is really that prism that I talked about before. It's the one that produces things like rainbow effects from white light. And the echelette are the kind of making use of that metal sheet with lots and lots of lines drawn on it. So different wavelengths filters obviously exist. Absorption filters are those like kind of stained glass pieces of colored glass. Interference filters are also present. So these absorption filters will block light over a broad range, but they might not block all of the light. They might just block some of the light. So what they can be useful for is actually just reducing the intensity of the light. Sometimes the light that's produced from our radiation source is too strong and it can damage our sample too much to get a meaningful measurement from. And so we need to reduce that intensity somehow. So one of the ways that you can do that is passing it through an absorption filter where some of the light is absorbed by the glass itself. So absorption filters are the simplest. So we have colored glass, for example. We have combinations that can cover reasonable spectral, but only in the visible regions. So like you can combine like a green filter with an orange filter to try and cover different parts of the spectrum that you want to cut out. And they often have high absorbances over non-active ranges. So what does that mean? That means like so say we just want to block out light at 500 nanometers. We're also blocking out all of the light here and here, even if we don't want to. It's still happening. It's still being blocked out. The problem with absorption filters is that they're time consuming to use. So like, for example, you have to manually change the filter in use. You have to have a box of filter glasses that are available. You have to change them one by one. So it's easy and it's cheap, but it's also time consuming. So that's why like things like monochromators are more common and they are contained within a lot of the spectrometers. So combining filters, so you can combine filters, like for example, we have a green absorption filter and we have an orange cutoff filter. So cutoff filters cut off all of the light beyond a certain region. So for example, this orange cutoff filter, it cuts off all of the light below 500 nanometers, but basically allows the transmittance of all of the light above 500 nanometers. So two filters can produce a narrower band of light, and combining a cutoff filter with an absorption filter results in only the common wavelengths being transmitted. So the combination here is a less than, so for example, the light that might have been transmitted by the green light or the green filter is cut off because of the presence of this orange cutoff filter. So all of this light is cut out. Beyond that, then the orange filter allows most of the light to pass through, but except here in the green part where the intensity is reduced and a lot of it is absorbed and removed. So cutoff filters typically have approaching 100% transmission over a select range and 0% over the rest. That's, I mean, it's not immediate, but like you can kind of see that it starts to allow a lot of the light to pass through. Monochromators then, so these are much more selective than filters or even combinations of filters. Like you can select very narrow wavelength ranges of like less than 1 nanometer. They're very accurate. They produce very, very narrow wavelengths of light or wavelength ranges. So we can really measure at a particular wavelength. Then they make use of windows, slits, mirrors, lenses, and prisms or gratings to select wavelengths over a wide range. So like what this means is that like they have a wide working range. So pretty much over the entire spectrum, they can select different wavelengths of light. So like there's no problem, like the same monochromator can measure light maybe at 100 nanometers, at 200, at 300, even 1,000 nanometers. But every time it measures, it's only really measuring that wavelength. So it can tell the difference between like 100 and 102 nanometers. Like it can tell the difference between those two different wavelengths and the light that's absorbed there or the light that's emitted there. And so they're widely used in modern spectrometers, but they are quite expensive and they really drive up the cost of spectrometers. But they're really, really useful. And like they don't involve really any user manipulation. Like the instrument does everything using the inbuilt computer controls. And so it can select the wavelengths automatically. So we don't have to like mess around with filters or something like that. So this is the simplest of the monochromators, which makes use of a prism. So many older instruments make use of prisms because it was very hard to make those like sheets of metal with gratings on them that I talked about before. So say we have 2 wavelengths of light entering here through the entrance slit into our monochromator. It doesn't look exactly like this. It'd be in some sort of box, but anyway, so the general flow is this, where the 2 wavelengths of light come in through the entrance slit, and then they're dispersed because of the size of this. Think of the double slit experiment. And then that is dispersed onto a culminating lens, which parallelizes the beam of light, makes it more coherent. Then this is focused onto a prism where the light passes through at different rates because of refraction, which we learned about in the previous session. So the light has different wavelengths, and so will pass through at different rates. And so that some of it will be bent more than others. And so that kind of splits the light. So the light gets split into its 2 wavelengths, wavelength 1 and wavelength 2. So wavelength 1 is in gray, wavelength 2 is in blue, but that's kind of arbitrary. And then we have, after that, the beams are focused onto the exit slit. And so the focal plane of the exit slit is where the light is focused. But you can actually move the exit slit. So for example, we want to measure wavelength 2 right now, but we could move this slit over here and we could measure wavelength number 1. And that's how the monochromator works. Basically, we disperse on the exit slit focal plane the different wavelengths that we want to measure. And then we can just move the slit to allow the light that we want to pass out. And then it would come out here onto, like, for example, our transducer or whatever, and it would get converted into some sort of photoelectric signal. So basically monochromators, the simplest ones make use of a prism to split multiple wavelengths of light. Okay, so the next part then is the sample cell that we'll be looking at, trying to see different materials which are usable. So we already mentioned about that whenever we talked about quartz and things like that. So the sample containers, typically we come across sample containers that look like this, which are called cuvettes. These cuvettes, look like plastic cuvettes. They can hold our sample. We can empty them out and reuse them again, so long as they haven't been destroyed by the presence of our analyte. They hopefully will be transparent over the range of the measurement that we're making. So sample containers and optics with cuvettes, with lenses, with prisms, gratings, filters. So we're mostly using cuvettes here, although you can get like glass windows and then sandwich your sample between the glass windows. So that's what you use whenever you're using like sodium chloride as your material. So suitable sample container materials are glass for 400 to 300 nanometers. So that's the visible and near IR. Silica quartz, which is usable in the full range of UV. So, and also most of the IR. I talked about before, silica quartz is kind of like the gold standard, but it is really expensive. The reason why it doesn't go above 3000 nanometers is because it actually starts to absorb. So silica glass is made up of similar materials to what we saw in electrochemistry. Those glass electrodes for the pH measurements. That's kind of the structure of the silica quartz. This is just one form of it. So eventually the silicon and oxygen start to absorb light, but they don't over this range. Then sodium chloride is very good as well, but the problem with it, of course, is that if it gets in contact with water or even the room is very humid, it can destroy our precious sodium chloride windows or sample containers. And so this is what the inside of a UV-vis spectrometer might look like. So you've got a sample holder here, and then the beam of light will pass through it, like so. And then the rest of the stuff like the monochromator, the light source, everything else is inside this white part, which extends around the sample. And so we don't typically interact with that. So here are some courtesy of one manufacturer of these sample containers called Helma, and their different materials that they have. So silica glass is good to about 350 to 2000 nanometers. So that's visible and near IR, definitely very usable glass for visible range. Quartz and fused silica is good to about 190 nanometers. So if you need to measure in the UV, that's what you're going to be needing to use. Plastics, visible only, but the solvent choice is important. Why is it important? Because some of the plastics will dissolve in our in certain solvents. So if you're using organic solvents, some of them may interact with the plastic and melt it. So it might seem okay whenever you're filling it up and then put it in the spectrometer. But when you go back later on to measure the thing, you may have a spectrometer full of melted plastic and your sample, which is going to be a big problem. Or what can happen is that it kind of interacts with the sample container. Maybe it doesn't dissolve it entirely, but it kind of infuses into the walls of the plastic and makes it go all cloudy, which also ruins your measurement and also your sample. So you can see here different things. So we have things like they're different materials, some of them brand names, but you can see the transmission and the amount they transmit here. So it's not completely uniform. It's not like, at 150 they stop absorbing. It's gradually they stop absorbing, but there is always a certain amount of absorption which happens, just because it actually the glass is there, and so it will absorb some of the light. We have, for example, optical glass here, which is good from about 300 or maybe slightly less than 300, and then beyond about 2500 it's pretty much useless. And here you can see some of the different types, like there's some long path length ones, some short path length ones, some which have like, they're blacked out. And then they have just different setups. So that the reason why some of these are blacked out is because they might fluoresce. And so maybe you want to avoid that fluorescent light interacting with your photodetector or something like that. Okay, so that was a sample cell holder. So typically for UV vis measurements, you want to be using either plastic, glass, or quartz. Quartz is the best, but expensive. So glass is usually where you reach a compromise. Plastic is really only for very specific instances. All right, the photoelectric device. So this is how do we detect our actual light? So this is the last thing we'll talk about in our discussion of the optical instruments. So ideally a radiation transducer, which is what is converting our photo signal into an electrical signal. Ideally, it is highly sensitive. It has low noise. So what does that mean? That means it doesn't produce a lot of background signal, so that the signal is predominantly or totally, hopefully, from the measurement of our sample. Highly sensitive means that it doesn't lose much of the light. So like any light which is incident on it, no matter how intense it is, will trigger a response from our, so these are kind of intertwined. So we want it to be highly sensitive, but we don't want it to be sensitive to things which are not our measurement. We want a wide wavelength response. Why do we want this? Well, we want this because we want to be able to use it over multiple different wavelengths. It's not very good if it's only usable over a narrow range. Like, how do we use it for different samples? It should have a linear output. So the signal that is received should be proportional to the intensity of the light which is incident, and it should have a low dark current. So the small current when the intensity is zero. So really, hopefully we're not measuring anything whenever the intensity is zero. So this equation becomes this equation where we have the signal is equal to some constant times the intensity of the light, which is incident on the transducer, plus the dark current, which is also proportional to some sort of instrument constant. So this is like basically like how fast it responds to the light, more or less. Okay, so some radiation transducer. So over here we have like a phototube. So this converts radiation to electrical signal. Dark currents are common, so these are signals without radiation. And typical photon transducers are here. So we have photovoltaic cells, we have phototubes, we have photomultiplier tubes, otherwise known as PMTs. We have conductivity transducer, silicon photodiodes, and charge transfer transducers. So I have labeled in red the ones which are most common. So there are 6 types. The most common are PMTs, silicon photodiodes, and charge transfer transducers. So the ones which are most common maybe in UV vis spectrometers are these photomultiplier tubes, which are the same as phototubes. They're just more of them. So like if a phototube can detect light, what they did was they got a lot of phototubes, put them all together and made a photomultiplier tube. And so PMTs, we'll discuss those in the most detail. The rest we won't discuss because they're not that common. But basically what we're doing here is, as I've tried to express on you as much as possible, we measure, actually what we measure in what we record and what we use to get our signal is electricity. What we want to measure is absorption, but that's quite hard. So we measure it kind of, what do you call this? Indirectly. So yeah, so photomultiplier tubes. So photovoltaic cells. So these are some sort of like descriptions. So the photovoltaic cells, the photon generates a current at the interface of a semiconductor and the metal. So photovoltaic effect, you might have learned about in Gen Chem 1, general chemistry, sometimes it's talked about there. I think I talked about it before. This is where like the light or the radiation or the photon packet interacts with the surface of the material and creates an electrical current. The phototubes then, so a photon causes emission of electrons from a photosensitive solid surface. So again, so the light is incident on the surface of some sort of material, and it ejects electrons from that. Those electrons can then be detected and create an electrical current. Photomultiplier tubes, we'll talk about in the next slide. Photoconductivity transducers, so photons absorbed by a semiconductor produces electrons and holes, enhancing conductivity. Then we have silicon photodiodes, so photons create photons create electron-hole pairs across a reverse biased p-n junction. So this is to do with our semiconductors, which we talked about before in the electrochemistry part. Then charge transfer transducers, so photons absorbed by silicon crystal create charges that are then measured. So as again, I said before, the most important are PMTs. The rest you should be aware of, but the most important one that we will talk about, and the one which is probably the most useful and most used in the measurements that we'll be talking about, are PMTs, or photomultiplier tubes. So here is a little bit more detail about the photomultiplier tubes. So the reason why they're so useful and so widely used is because you don't actually need that much light in order to get a reasonable measurement. So they're suited to low power radiation because basically they get that low power radiation, and then through a series of internal processes, they generate a lot of current from that. So it actually needs to be protected from high power because that can cause irreversible damage. So these need to be kept in the dark, which is why the enclosure of like a UV vis measurement instrument is quite, it's really enclosed, because if too much stray light from the room, for example, got in there, it would literally destroy the photomultiplier tube, because there's so much light is incident on the photomultiplier tube, and it amplifies that light. It just blows it out. So these are similar to phototubes, but with diodes, basically extra electrodes, which generate more electrons. So there's a photocathode material, which emits electrons when irradiated. It's usually made from gallium or arsenic. And then the process converts one photon into around 1 million, or maybe 10 to the 7, 10 million electrons. Then the PMTs have large dark currents, but this can be limited by cooling to about minus 30 degrees Celsius. So usually like the most sensitive PMTs cool down to minus 30 degrees Celsius. So this is one of the reasons why whenever you turn on a UV vis spectrometer, sometimes you have to wait a little while so that there's like a heat exchange plate inside of the instrument can kind of cool down the PMT. Although a lot of UV vis measurement instruments are now moving towards things like CCDs as opposed to PMTs, but PMTs are still very useful. So this is a schematic of one. You don't really need to know how to draw this, but you do need to be aware of the process which is happening. So the light is incident on a photoemissive cathode. So the light comes in and hits this material, which generates an electron. These electrons then are attracted towards the diodes. Whenever they hit the diode, they produce more electrons, so several electrons from each incident electron. So like, for example, one electron goes from here to here, it produces 9 electrons. Those 9 electrons then go from D1 to D2, produce another 9 electrons each, and so on and so forth. So every time you're producing many, many more electrons, until such times as you produce so many electrons that you have around about 10 to the 7, more than 1 million electrons have been produced for one photon. So if you had 10 photons, you would have 10 million electrons produced. So that's why it needs to be protected from a lot of incident radiation, because otherwise you just end up with so many electrons that the thing would just have too much energy and just be destroyed. Okay. And so that's kind of our discussion of the optical components done. Rather brief, we didn't go into too much detail on everything, but more detail will be discussed in your future instrumental chemistry classes.